of different platforms, because that gives you a fundamental basis for understanding how they work. So, first, we'll start with the fact that making libraries, for those of you who may have in the past done clone-based libraries for capillary sequencing, is now faster, easier, and cheaper than ever. There's no need to run through an E. coli intermediate. There's no need to do cloning. It's all a very straightforward process that begins with the random fragmentation of the starting DNA that you're interested in sequencing. And if, for example, these are just PCR products and there's no need for fragmentation, you can just go forward to the next step, which is ligation of these fragments with custom linkers or adapters to make a library. And as you'll see with each one of these technologies, library construction is basically the same approach. Each instrument has their own specific and unique adapters, as you might guess, but nonetheless, the overall process is exactly the same and highly concordant. So instead of spending a week producing a subclone library, which you then pick, amplify E. coli, and isolate all of the DNA in the old-style process, instead with this process, you essentially, in the period of a day's time, fragment the DNA, add on the adapters by ligation, do some purification and amplification steps, quantitate the library, and you're ready to go. So the whole process literally takes less than a day's time. It costs in our hands on the order of about $100 or $150 to complete, to the point where you now have hundreds of millions of DNA fragments that are ready to do next-gen sequencing on. So I mentioned that there's library amplification in these processes. Depending upon the platform you're talking about, it takes place on some sort of a solid surface, so either a bead or a glass surface, and I'll show you the differences between those different sequencers. But the net impact is the same. You're taking these unique fragments, and now you're starting from one fragment, amplifying it up to multiple copies. Well, sometimes when I present this lecture, I ask students, why do you think we do that? I won't do that for you guys, but I'll just tell you the answer, which is that for all but some of the single molecule sequencers, which I'll mention as we go through them, you must do library amplification in order to see the signal that's coming back from the sequencing reaction itself. So most of these sequencers start with single molecules. They're amplified in place, either on a bead or glass, and then they're sequenced. And to see the sequencing reaction going on in real time, you actually have to do that amplification step. So that's not a bad thing in most cases. However, with any type of enzymatic amplification, you're always going to get some aspects of biasing and some aspects of what's called duplication or jackpotting, where some of the library fragments will preferentially amplify, and you get more of those sequences than of others. And so we have ways to adjust or ameliorate for that in our processes. Okay, and then onto the sequencing reactions themselves. For most of these technologies we'll talk about today, a direct step-by-step detection of each nucleotide base incorporated during the sequencing reaction occurs. Commonly, these approaches are referred to as sequencing by synthesis, if you will, if they use a polymerase, or sequencing by ligation, but they do occur in a direct step-by-step fashion. So again, let's harken back to days of old in capillary sequencing. If you're familiar with this, what happened procedurally was you sequenced all of your DNA fragments, perhaps 96 or 384 at a time, and then you applied them to the sequencer after the sequencing reaction was over with, and they were separated by electrophoresis, and the fragments were detected, either by radioactivity if you're as old as I am, or fluorescence if you're younger than I am. So, in contrast, next-generation sequencing, everything happens together at the same time. Sequencing and detection happen in a step-by-step fashion, so that you essentially don't decouple the sequencing reaction from the detection of the sequencing reaction. And this leads to another name, which I actually quite prefer over next-generation sequencing, or some generation of sequencing, which more accurately represents what's going on in these sequencing instruments, as I hope you'll come to appreciate. Namely, you're performing hundreds of thousands to hundreds of millions of sequencing reactions all at the same time. And so the term that's often applied to these technologies is massively parallel sequencing, which is exactly what you're doing. You're sequencing everybody together simultaneously, performing an imaging step to detect what happened, and then moving on to the next base incorporation step over and over and over again until you generate your full sequence run. Now, the consequences of doing next-generation sequencing, with a couple of exceptions, again, that I'll point out, is that in general, these reads are shorter than capillary sequencers. And there are a number of reasons for this, but it mainly comes down to one word, signal versus noise. I guess that's three words technically, although I think of them as one. So what you're always battling in this detection game is the signal-to-noise ratio, and in most of these technologies, there's some cost to pay that ultimately limits the read lengths. And I'll give you the specifics for each platform, but just consider in principle that these are going to be shorter read lengths. And I mentioned earlier the contrast between the cost to generate data and the cost to analyze data, and this is where push comes to shove, because there's a toll exacted from the fact that you can produce lots of short reads, and then you have to go and analyze those. And I'll point out to you why the reasons why that becomes much more difficult, and why the bioinformatics overhead, the analysis overhead, is quite expensive for us still. So I already alluded to this for you, but I'll talk about it again a little bit later on when I show some examples of how we're actually using now the fact that these are digital reads. So each read of a massively parallel sequencer originates from one fragment in the library, even though it's amplified. What that means is you can literally apply counting-based methods to the analysis of these data that will tell you things, for example, as I'll show, how many tumor cells in the collective that produce DNA for a tumor genome actually contain each one of the mutations that you've detected. So you can get it down to that level of sensitivity. You can look at the number of counts for a given messenger RNA, for example, and look at quantitative aspects of sequencing as we've never been able to do before. And this is a tremendously exciting application application space for next-generation sequencing. I'll try and give you a feel for that. And then lastly, one of the newest abilities that's come on board for these sequencing instruments is the use of what we refer to as paired-end reads. So most of these technologies started out by priming a sequencing reaction, extending off of a single primer for a certain read length, and that was it. And you got a single fragment read. And in most cases, that was pretty darn good, and we learned to work with it. But over time, what emerged was the ability to sample from not one, but both ends of the fragment in that library. Namely, each technology applies a different adapter to each end of the DNA fragment that's being put into the library, and you can exploit that by using one primer for one adapter, and in a second round of sequencing, a second primer for the second adapter, effectively collecting data from both ends of the fragment, and also understanding that based on the size of fragments that went into the library that you made, let's say 300 base pairs, now that you have 100 from one end and 100 from the other end, you can actually align those back to a reference genome and expect that they will align at about 300 base pairs apart from one another. And when that doesn't work out, and you have a distance further apart or closer together, or maybe even mapping to two separate chromosomes, you can actually use that information to make sense out of the genome that you're sequencing. And I'll give you a few examples of this from our work a little bit later on. So paired-end reads had all kinds of other advantages that I've listed here. There's also a bit of a nuance to paired-end reads that I want to spend a little bit of time on because it is a major point of misunderstanding, if you will, in various literature, and manufacturers, quite frankly, will try to trip you up with this one. So you can get the paired-end read data. Sequence can be derived from both ends of the library fragments, as I just mentioned. There are basically two kinds of paired-end reads, however, that go by different names. So in my vocabulary, true paired ends mean that you have a linear fragment. It's typically, as I said, on the order of 300 to 500 base pairs, if you will, and you're going to be literally using two different primer extension steps sequenced at both ends in two separate reactions. So that's paired end reads. The second type of read pair that you can generate on a next-gen sequencer is a so-called mate pair. And the nuance here is that rather than using two separate adapters, you literally circularize a large DNA fragment, typically greater than a kb in length. 3, 8, 20 kb libraries are typically made. And by circularizing that around a common single adapter, you actually can generate mate pairs where the ends of the DNA come together. You go through a second step to remove the extraneous DNA that's the part of the circle you don't care about, and you either use the adapter to sequence across the DNA, or you do a single reaction read, use a single reaction read to sequence across the DNA, or two separate end reads to tell you the sequences at either end of those fragments. So the advantage of mate pairs is that you can stretch out to much longer lengths across the DNA that you're interested in sequencing, and hopefully understand better the long-range structure of that DNA. The downside of mate pairing as opposed to paired end reads is that this approach, because DNA circularization is inherently not very efficient, is that large amounts of DNA, typically several micrograms, are required for each library that's constructed. Okay, and that just goes to the inefficiency of mate pairing. So in general, whether they're mate pairs or paired end reads, these offer advantages for sequencing, especially when the genome is like the human, large and complex. The reason why is you can more accurately place that read on the genome than you can a single-ended read, and the main reason for that is as long as both reads don't fall into a repetitive structure, you can anchor one with certainty, even if the other one doesn't anchor with a high degree of mapping quality, as we call it. So it may be a read that could place at multiple positions in the genome, but as long as the companion read to it places exactly at one place in the genome, when you go back to align the reads to the reference, you can identify where exactly that read came from. So the net result of this is that you can use more reads towards your ultimate analysis than you might be able to do with just single-end reads, and this provides a huge advantage in the economy of sequencing as well. Okay, so that's the sort of introductory similarities and differences and basic terminology. Now what I want to do is use those concepts to walk you through each of the different types of approaches and keep those similarities in mind as we also examine the differences. So this was, as I mentioned, one of the first technologies to come to us for next-generation sequencing around about late 2004, early 2005. This is a massively parallel version of a sequencing approach you might have heard about called pyrosequencing, which basically uses the emission of light to register the incorporation of DNA nucleotides into a growing a strand. So the 454 approach to constructing a library is exactly as I walked you through. So we do a random fragmentation step. You can see the DNA is intact here, now in small pieces, now with the adapters ligated onto either end, and actually this is a denatured molecule, which is the precursor to the amplification step. So you take your single-stranded adapter-ligated library now. So the yellow are the DNAs that you care about. The different colors at the ends are the specific adapters for this platform. And you essentially do a step called emulsion PCR. This is the amplification step that I talked about earlier. And it's a really unique way of doing this. Namely, all of the PCR occurs in an emulsion of oil and reagents that are in aqueous phase. So if you can see this hopefully well enough, what we try to achieve in emulsion PCR is a micelle, a sort of clear area in the center of the picture, that contains a bead, shown here, for amplification. And if you can see the little squiggles on the surface of the bead, those represent the complementary sequences to the adapter that you ligated on in your library construction step. And for each amplification, ideally we have only a single molecule fragment from that library in association with the bead. Now, what's invisible in this aqueous micelle, of course, are the basic building blocks of DNA, as well as DNA polymerases that are going to effect the amplification of this single fragment on the surface of the bead during a PCR step, namely temperature cycling. Of course, we aren't just doing one bead at a time. We're doing several million beads at a time in a single microtube in this oil and water emulsion called emulsion PCR. Okay? So you go through the PCR cycling steps. What you end up with is this micelle containing a bead with lots and lots of copies of this